Okay, so in this video, we're going to be talking about heme metabolism. So under heme metabolism, we're going to look at heme synthesis as well as the disorders that are related to heme metabolism. We'll also learn about heme degradation and its disorders, for instance, hyperbilirubinemia. Okay, so firstly, to start with, what exactly is heme? So heme is a derivative of a porphyrin. Okay. Now these porphyrin, they are cyclic compounds. All right. So this is just a brief example of a cyclic compound. All right. This is what a porphyrin is. So this cyclic compound is formed by fusion of four cyclic compounds. So 1, 2, 3, 4. Actually, let's delete one. All right. So it's a fusion of four cyclic uh, compounds that we call pyro rings. So these are what we call the pyro rings. Now these rings, they are linked by a special bond that we call methynyl bridges. These are what we call the methynyl bridges. Let's go direct into the synthesis of heme metabolism. Okay, so what you notice is that heme is a prosthetic group of hemoglobin, okay, of hemoglobin as well as myoglobin, cytochromes, catalase, peroxidase, as well as tryptophan. Okay, so a prosthetic group determines how that compound is going to function. So to start with, heme is synthesized in almost all mammals, in almost all mammalian tissues, this is to say. Alright, so to start with the organelles where heme metabolism occurs, one, we have the mitochondria as well as the cytosol. Okay, so synthesis is most active in the bone marrow. Okay, knowing that it is in the bone marrow where red blood cells are formed, so synthesis is most active in the bone marrow for the incorporation of this for the incorporation into the hemoglobin as well as the cytochromes respectively. So to start with, in order for heme synthesis to begin, the first products that we have are succinyl CoA and glycine, and all this is happening in the mitochondria. So firstly, glycine combines with succinyl CoA. Okay, now when this reaction occurs, the end product is aminolevenic acid, which is this, or commonly known as ALA. So that's the product, and the enzyme that's used is aminolevenic acid synthase. All right, now this is the first reaction of the heme metabolism, and it being the first reaction, this is the rate limiting reaction. So whenever you hear rate limiting reaction, this is to tell you that it's at this reaction in the heme metabolism where regulation is done. It's either it can happen or not, or rather it can be inhibited or not. It's at this reaction. Alright, so this is the product, the ALA, and in as much as there's the enzyme, the aminolevenic acid, there is also another coenzyme that we call pyrophosphate. Okay, so we have pyrophosphate, or commonly PLP. So pyrophosphate is the one that serves as a coenzyme for aminolevenic synthase, which is the enzyme. So if it's a coenzyme, meaning it's helping to enhance the catalytic reaction, or rather catalytic power of that enzyme. So the next thing is that LA leaves the mitochondria. Remember, all this reaction was happening in the mitochondria. The first one, we had glycine and succinyl-CoA combining to give us ALA. Now this ALA leaves the mitochondria and goes into the cytosol. All right. Now in the cytosol, two molecules of ALA, which are right here, they combine, and their combination, or rather when they condense, they form what we call porphobilinogen. All right. And in the process, two molecules of water are released. All right. The enzyme that's used in this case is porphobilinogen synthase. And another thing to note about this stage is we have an enzyme that we call LA dehydratase. This LA dehydratase, it's a zinc containing enzyme, and because it has zinc, it's highly sensitive to heavy metals like lead. Okay, so when there is lead, it can lead to the inhibition of this enzyme, which is ALA dehydratase. Okay, so when you look at this porphobilinogen, which is synthesized, it's the first pathway intermediate that includes a pyroring. Okay, in all these intermediates, this porphobilinogen is the first one that forms a pyroring. So take note. So the next reaction is, now that we have porphobilinogen, remember we had one pyroring. So 4 pyrorings are going to come together, right? There will be a formation of a tetrapyro, meaning 4 pyrorings. They come together, they condense into 4 molecules of porphobilinogen, and they form a linear tetrapyro that we call hydroxymethylbilane. This hydroxymethylbilane is composed of 4 pyrorings of porphobilinogen that are fused together. And the enzyme that aids this is porphobilinogen deaminase. This is the one that's making all this happen. So as we have hydroxymethylbilane, what happens is that another reaction happens. So this hydroxymethylbilane, it cyclicizes, and it's converted to uroporphyrinogen, which is this uroporphyrinogen 3. Alright, so there's just a shifting. So when you look at this hydroxymethylbilane, of course we have 4 pyrorings, but there's a point at which they are not fully conjoined. Right, now uroporphyrinogen 3 is where, when this reaction occurs, now the 4 pyrorings are conjoined together. That bond is formed and water is released, like you can see. So this uroporphyrinogen 3 is converted to coproporphyrinogen 3. Alright, so there's a conversion from one molecule to the other. And then what's happening here is decarboxylation. So this is to say that uroporphyrinogen 3 is being decarboxylated to coproporphyrinogen 3, and water is being released in the process. Now the enzyme that's involved is uroporphyrinogen 3 decarboxylase, like you can see over here. So the next reaction will automatically start with coproporphyrinogen 3. So coproporphyrinogen 3, it then enters back into the mitochondria. Remember that all these processes were happening in the cytosol. Now, when we reach the stage where coproporphyrinogen 3 is synthesized, it goes back to the mitochondria, where it gets oxidized by coproporphyrinogen 3 oxidase. Okay, when it gets oxidized, it's converted to protoporphyrinogen 9, which is this chemical element right here. And the enzyme that is involved is this one that was earlier mentioned, porphyrin 3 oxidase. All right. So we're almost getting close to finishing. Now that we have protoporphyrin 9 in the reaction, this protoporphyrin 9 is oxidized further to protoporphyrin 9. Okay, notice the change in names. Protoporphyrin is now changed to protoporphyrin. Okay, it's just like in the stomach during digestion, where you have the body stores pepsin as pepsinogen, right? So meaning pepsinogen is inactive. Now when pepsinogen is activated by HCl in the stomach, it's later turned to pepsin, of which pepsin can now work on the proteins. So the same thing is happening. Protoporphyrin 9 is the inactive form of protoporphyrin 9. So this is what's happening, and the enzyme that's involved is protoporphyrin oxidase. Remember, whenever we have an oxidation reaction, you can notice from the name of the enzyme that this is what's happening. Okay, then protoporphyrin that was formed earlier on, an ion is added onto it. Okay, and that ion is ferrochelatase. That enzyme that aids the addition of that ion to protoporphyrin is called ferrochelatase. Okay, and this is the final step in the heme synthesis, because now we are incorporating the ferrous ion into the protoporphyrin ring. Okay, after having made those 4 pyro rings, and after it's active, a ferrous ion is added to it, and the enzyme involved is what we call heme synthase. Okay, so this is the final stage where now we finally have our heme B. So this is exactly what's happening. So next up, we are just going to look at the regulation of heme synthase. So there are several reactions that have been skipped. Okay, so this is just to show you exactly what's happening. So regulation starts with where we have glycine plus our succinyl-CoA, giving us our aminolevonic acid, which is ALA. Alright, now remember we said this is the rate limiting step of the reaction, right? So because it's the rate limiting step of the reaction, what you notice is that ALA synthase, which is the enzyme that's used to convert this reaction to this, it's inhibited by what we call hemine. Hemine is a compound. Hemine is a compound that inhibits this enzyme from working, as well as we also have this LA dehydratase and ferrochelatase, which is here. Okay, so these two enzymes are inhibited by lead instead. Alright, so lead poisoning results in high levels of ALA. Now this ALA shouldn't remain in its state. In this state, the original state, is supposed to be converted to other things. Otherwise, when it accumulates, it's very toxic for the brain. That's why it's dangerous. Okay. The next up, we'll just look at some of the disorders that are associated with heme biosynthesis. So these inhibitors that we're going to talk about, or rather these disorders, we call them porphyrias. So genetic disorders that are associated with deficiency of the enzymes that are involved in the heme synthesis are the ones that we are calling the porphyrias. So these, in order for you to see the symptoms of that disease, you see it in blood, you see it in urine, you also see it in stool. Okay, so there are different types of porphyrins, and this all depends where they accumulate. Okay, and because of these porphyrins, they can cause nerve and skin problems. And some of the other causes that are known, or rather that are found, they can be passed on from parents to from parents to their children. We call them hereditary because they can be passed on from parents to their children. Okay, so they can either be autosomal dominant, okay, meaning this disorder is found in other cells other than the sex cells. So they can be autosomal dominant, or they can be recessive, or they can even be X-linked, meaning they can be found in the chromosome that determines the sex of, or that determines the gender of that person. Okay, so they are triggered by various things, or rather um, by various factors. Okay, some of the factors that you encounter are: one, we have alcohol, too much of alcohol consumption; two, smoking; three, sunlight; four, stress; then five, certain medications; six, some eating disorders; seven, HIV infection; eight, lead accumulation. Okay, and so on and so forth. So these are some of the things that can lead to some of these porphyrias. All right, so the two major ones, the two major types, are what we call one. Oh, sorry, let me just write it nicely. So one is cutaneous porphyria. Okay, so these are cutaneous because they affect the skin, and some of the symptoms that can be seen is oversensitivity to sunlight; two, shrunken gums; three, we also have blisters on the skin. So these are some of the things that these are some of the symptoms that you are able to pick. Okay, and a person that has these signs, they often have itching, or rather swelling, and so on and so forth. So the other type of porphyria, we call them the acute porphyrias. Okay, now these porphyrias, they affect the nervous system. Okay, so those who are affecting the skin, these are affecting the nervous system. Now some of the symptoms include high blood pressure, irregular heart rate because of this, constipation, problems in breathing, and so on and so forth. So next we are just going to now look at the actual disorders and the enzymes that they're affecting. All right, so the first one that we'll talk about has to do with the synthesis of the aminolevinic acid. Okay, and we know that the enzyme that's involved is ALA synthase. Now whenever this enzyme is defective, the disease that's caused is called X-linked sideroblastic anemia. Okay, and the other one is X-linked protoporphyria. The second one, we have ALA dehydratase or BPG synthase. Okay, and this enzyme is found where we have formation of porphobilinogen. Remember, when we have two molecules of ALA, they give us porphobilinogen. Now, when this enzyme is defective, what you have is LA dehydratase porphobilinogen. So like you can see, the name is going with the disorder. LAA ALA, sorry, dehydratase porphyria. Okay, then the next one is BPG deaminase. This BPG deaminase, the disorder is that it results in what we call acute intermittent porphyria. Okay, so this is a condition that's involved. The next is UPG 3 synthase, and this results in congenital erythropoietic porphyria. So these porphyrias are very easy to learn, and they should be on your fingertips as you watch this video, as you study your notes or take down your notes. Make sure you know the names and what enzyme is being affected. So the UPPG decarboxylase, the condition is porphyria cutanea. That's the congenital disorder. The other one is CPG oxidase, which is hereditary coporphyria. The other one is protoporphyrin oxidase. When this enzyme is affected, what we have is variegate porphyria. And then this one is ferrochelatase. Remember the last step of heme synthesis. Whenever we have that, we later on have what we call erythropoietic protoporphyria. That's the disorder that's formed. Alright, so next up we are going to look at what happens in mutations of DNA. Okay, so just draw a brief table. So when the DNA is mutated, there will be abnormalities with the enzymes that synthesized heme synthesis. So you are going to have abnormal enzymes. Okay, when you have abnormal enzymes, you what will happen is that there will be an accumulation of ALA as well as PBG. All right, you also have what we call accumulation of the porphyrinogens in the skin. Okay, porphyrinogens in skin increase, or because of DNA mutation. There is also spontaneous oxidation of those porphyrinogens to porphyrins. Okay, so those porphyrins are going to be highly active, and if they're active, they'll be the person is going to be photosensitive, so they'll be sensitive to light because of that. So next up, we are going to look at heme metabolism disorders. Okay, now under this, let's look at the fate of the RBC. Okay, so RBCs, the lifespan that they have is 60 to 120 days. After this, they must be degraded. Now RBCs are lysed. They're supposed to be lysed or broken down, and the breaking down starts in the ER, which is the endoplasmic reticulum of the liver. These are the organs where they are phagocytosed: the liver, the spleen, the bone marrow as well, and the skin. Okay, now when RBCs get broken, okay, or rather when they get broken down, what comes out of them is 1. globins, 2. heme, 3. iron. Okay, so it breaks down to its initial the things that make it up. Okay, and in him we have bilirubin. So heme is converted to bilirubin. Globins are converted back to amino acids in amino acid metabolism, whilst the iron is transported to what we call transferrin, and then it's used in the next heme biosynthesis. So it's recycled after it's broken down. Alright, so the heme, like we had said in the first place, when we have this red blood cell, the red blood cell is broken down, heme is released. This heme is converted to what we call biliverdin. Okay, biliverdin is later on converted to bilirubin. Alright, now bilirubin metabolism that's normal is going to look like this. Okay, so we have feather fate of the bilirubin that's formed. So what happens is that it can be divided into several steps, and remember that the metabolism, or rather the excretion of bilirubin, occurs in the liver as well as the intestines. So what happens to this bilirubin? Firstly, there is uptake of bilirubin by the liver. So it goes through the liver, then it undergoes through what we call conjugation. So it's conjugated, it's added on to something else. After conjugation, bilirubin is secreted. So it's secretion in bowel. So it's added to bowel. Okay, then bowel, remember, is added to, of course, food that has lost some water, and that's on its way to excretion. So it's later on excreted via feces as well as urine. That's how bilirubin is gotten rid of. So now, when there's a problem with the levels of bilirubin, you have a condition that we call hyperbilirubemia. Okay, this is a condition. So the normal concentration of bilirubin in a person is 0.1 to 1 milligram per deciliter. This is the total bilirubin. Okay, when it's conjugated, so let's say conjugated bilirubin, this is total bilirubin. Conjugated, it's 0.1 to 0.4 milligrams per deciliter. When it's unconjugated bilirubin, it's 0.2 to 0.7 milligram per deciliter. So hyperbilirubinemia is where the bilirubin in blood exceeds 1 milligram per deciliter, which is equivalent to 17.1 micromoles per liter. Alright, so whenever it exceeds, so if it's above, hyperbilirubinemia is when the levels of bilirubin are above 1 milligram. So this is the total. When it goes beyond this in the normal, it leads to bilirubemia. Okay, and this is also referred to as jaundice. If you've heard of a condition that we call jaundice. So in jaundice, you find people that have yellowish eyes or yellow skin because of this. So what are the causes of hyperbilirubinemia? One, when the production of bilirubin is very high. So this is bilirubin, high production of bilirubin. Then when the hepatic intake is low, meaning the liver isn't taking in as much as it is supposed to, okay? Then when it's not being conjugated, meaning it's not being added on to other substances. So when the levels of conjugation are low, so this is hepatic conjugation, okay? The other one is when excretion of bilirubin into bowel is low, okay? So excretion of bilirubin in bowel, when it goes down, that means the levels of bilirubin in the blood will increase. And if they're increasing, this is where you find the person has yellowish eyes, okay? So there'll be an accumulation of bilirubin in the blood. So the skin, the sclera, which is in the eye, will appear yellowish due to the deposition of bilirubin. Alright. So now jaundice develops when there's too much bilirubin in the blood, and we have three types. Type 1 is what we call the prehepatic. Okay, from the word pre means before, so it means this problem, it's a problem with the blood before it gets to the liver. For instance, malaria, sickle cell anemia, okay, and so on and so forth. Then while you have the hepatic, you can already guess that it's already reached the liver. So this one is a problem with liver, okay? This jaundice is when there's a defectiveness with the liver. So in cases of cancer or cirrhosis, which is caused by excessive drinking of alcohol. Then you also have post-hepatic, okay? Post meaning after. So this one is when the bile ducts are blocked, okay? So this can lead to what we call the gallstones, because now that those bilirubin is going to crystallize and is going to form gallstones. So in conclusion, jaundice is a symptom of a medical condition. It's not necessarily a disease. So when you see yellowing of eyes or yellowing of the skin, it's just a symptom that there's something wrong with how bilirubin is being handled in the body. Alright, with that said, that's the end of this topic.